In this video, I want to introduce you to spatial transcriptomics. We'll talk about what it is, why it matters, and how it leads into MERFISH, the specific method we use in our research. By the end, you'll be able to 1. Understand the differences between bulk RNA sequencing, single-cell RNA sequencing, and spatial transcriptomics. 2. See why knowing where things are expressed is just as important as knowing what is expressed, especially in the brain. 3. Describe the basic idea behind imaging-based transcriptomics methods like MERFISH. So, why does location matter in neuroscience? Imagine 2 neurons that express the same set of genes. If one is in the visual system and the others in the olfactory system, their jobs in the brain are entirely different. Spatial transcriptomics gives us the missing piece. It tells us not just what genes are active, but exactly where those active cells are within the brain's architecture. That's the key to linking molecular signals to function. Now, let's make this idea more concrete with one of my favorite analogies: berries. Bulk RNA sequencing is like making a smoothie from strawberries and blueberries. You can measure how much red and blue ingredient you have, but you've blended everything together. You've lost all information about which fruit came from where. Single-cell RNA sequencing is like keeping each berry intact, so you can inspect them one by one. You know exactly what's inside each berry, but they've all been taken off the plate. You can't tell where they were originally arranged. Spatial transcriptomics keeps both identity and location. It's like looking at the plate with all the strawberries and blueberries still in place. You can see exactly what they are and exactly where they are. This combination of identity and location is what makes spatial transcriptomics so powerful, especially for understanding the brain where organization is everything. There are 2 main ways scientists study gene expression in space: sequencing-based and imaging-based approaches. Sequencing-based spatial transcriptomics typically captures many or even all genes at once, but its spatial resolution is often limited by the size of the capture spots, meaning multiple cells can be mixed in each measurement. Imaging-based spatial transcriptomics, on the other hand, detects RNA molecules directly inside intact tissue sections using fluorescent probes. This preserves single cells and in some cases even subcellular resolution. MERFISH, which stands for multiplexed error-robust fluorescence. In situ hybridization is one such imaging-based method. It uses combinatorial labeling and sequential imaging to identify and count thousands of RNA molecules directly in their original positions within the tissue. By combining molecular identity with precise spatial location, MERFISH allows us to map the brain's architecture in its native context. In biology, cell arrangement defines a tissue's molecular architecture. In the brain, a neuron's position shapes how signals flow, networks form, and regions specialize. And it's not just the cells that matter, but also the genes they express. The spatial pattern of gene activity influences how cells function and interact. Spatial transcriptomics captures both molecular identity and physical location, revealing organization, boundaries, and interactions that would be invisible without spatial context. Next, we'll look at MERFISH, an imaging-based method that maps gene expression at single molecule resolution directly in intact tissue.